Sexual reproduction in flowering plants. Reproduction is one of the most important features of living organisms. For this purpose, angiosperm produce a special reproductive structure termed flower. Morphologically, flowers are considered as modified and condensed buds or shoots. Flowers possess reproductive parts and non-reproductive parts. This reproductive part is embryologically relevant, whereas this non-reproductive part is morphologically significant. Reproductive part means androecium, that is androecium, gynoecium. Non-reproductive part means calyx, corolla. These are non-reproductive parts. These reproductive parts embryologically relevant, whereas this is morphologically relevant, significant because calyx or sepals protects the flowers in bud stage. Right. Flower, a fascinating organ of angiosperms. Flowers are variously colored, shaped, and scented for promoting pollinating processes. Flowers are closely related to cultural, religious, social, love and affection, happiness, grief, and even mourning. Flowers are the source of various scents, perfumes, and dyes. Honey is a product derived from nectar of flowers collected by the honeybees. So, up to from here to here, nothing to be learned. It's just a general information, right? A typical flower has four whorls of the flower are calyx, represented by K, corolla C, androecium A, androecium gynoecium, that is G. The calyx and corolla are non-reproductive parts, as I've told you earlier. Androecium and gynoecium, spelling is wrong, are the respective male and female reproductive parts. All these four whorls are placed on the condensed tip of the pedicel known as thalamus. So here lies. So this is sepal. Above this petal may be there. This swollen part, this is a pedicel. Stalk of the flower is called pedicel. Stalk of the leaf is called petiole. Stalk of the leaf is called petiole. And this swollen part is called as thalamus, to which calyx is placed. This swollen part is called as thalamus, right? Stamen. The unit of androecium is a male reproductive organ. The unit of androecium is a male reproductive organ, and the carpel or pistil is a unit of gynoecium in the female is a female reproductive organ. Now, what is the unit of this calyx? Sepals. What is the unit of this corolla? Petals. Right. Now, this is pre-fertilization structure and development of male and female gametophytes. Male reproductive structure of a flower. Androecium is a male reproductive organ. It's an organ. And stamens are the unit of it. Each stamen consists of filament and anther lobes and connectives. So it consists of anther, filaments and connectives. Connectives lies here. Connectives lies at this spot in between that, right? Right. The connective is the extension of filament that binds anther lobes together and possess vascular tissues. Connective is the extension of filament It is an extension of filament and binds the anther lobes together. So anther lobe is like this means this is the connective. So this extension of filament, this is a filament, this is the anther. So this extension of filament into this bilobed anther. So this is called as connective. This is called as connective, right? And possess vascular tissues. So connective is this a very important statement. Connective is the extension of filament and binds the anther lobes together and possess vascular tissue. And possess vascular tissue. Right. Next. Anther lobes consist of four microsporangia. Anther lobes. See. So it's a bilobed structure. You call this as bilobed structure, right? When I cut it like this, when I cut and see like this, so it will be looking like this. Therefore, this is one microsporangia, this is one microsporangia. So one, two, three, four. So four microsporangia, or these are called as pollen chambers. Pollen chambers. Four microsporangia or pollen chambers. So what is haploidy? Ploidy, 2NN condition. Diploidy. Microsporangia, diploid. Okay. Each microsporangium, sorry, each microsporangium in turn consists of four wall layers and inner sporogenous tissue. Each microsporangium. So each microsporangium has four walls. We will see it one by one. Four walls surrounding it like this. And inner sporogenous tissue also. So it is called, the outer layer is called epidermis, followed by endothelium, middle layers tapetum. Middle layers tapetum. You observe this diagram. So as I've said you, how we are cutting this? We are cutting the anther like this. This is the anther. This is the filament. This is the connective. Connective is extension of filament into this anther and possess vascular tissues. So we are cutting the anther like this. If you see like this, it consists of four microsporangia. Four, one, two, three, four. Four microsporangia, or you can call it as pollen chambers. This is the anther lobes. And this is a TS means you're cutting it like this. TS means like this. LS means like this. You're cutting it like this. Anything like this means it is LS. TS means in this fashion, right? It's a okay. So outer this layer is called as. So you carefully observe this epidermis, followed by endothelium, followed by middle layers, and tapetum, followed by tapetum and sporogenous tissue. Sporogenous tissue. And this is a connective. You see this connective, right? Now you read the statement. The former two wall layers means epidermis endothelium provide protection. So this epidermis and endothelium provide protection and help in dehiscence of anther lobes and maturation. On maturation, this produces pollen grains. This has to be released out for pollination and further fertilization. Therefore, this has to get ruptured. This epidermis and endothelium help in dehiscence of anther lobes on maturation. Dehiscence means spreading out. Dehiscing means just bursting out and giving out the spores. Here pollen grains, right? Middle layer stores reserve food. Middle layer store reserve food. This layer store reserve food materials. Epidermis endothelium is for protection and dehiscence of anther lobes and for the dehiscence of the anther lobes on maturation, and middle layers store the reserve food. Now tapetum is the innermost layer which is nutritive in function. Nutritive to what? Nutritive to the sporogenous tissue, and the developing microspore. Okay, the sporogenous tissue are homogeneous. They are homogeneous means they composed of same kinds of cells, and on maturation these potentially act as microspore mother cell. The sporogenous tissue differentiate itself to microspore mother cell or pollen mother cells. We can call it as either PMC or MMC. Right. It's an enlarged view. You can observe this diagram: epidermis, endothelium, middle layers, tapetum. So tapetum is close to this microspore mother cells. Now a mature and dehisced anther. Dehisced means, see, this is the opening, right? This opening is called as dehisced. It is broken by epidermis and endothelium. Epidermis and endothelium. These are the matured pollen grains. These dot particles are the matured pollen grains, right? It's a broken wall layer. Now microsporogenesis. So microsporogenesis, as anther lobes mature, the cells of sporogenous tissue differentiated. As anther lobes mature, the cells of sporogenous tissue differentiate itself as pollen mother cells or microspore mother cells. And this, what is a ploidy? 2n. Sporogenous tissue, what is a ploidy? 2n. Diploidy. And this has to undergo meiosis, resulting microspore tetrads or pollen tetrads. You know that the sporogenous tissue is first developing into PMC or MMC. This PMC or MMC, that cell is undergoing meiosis. So meiotic first division, second division. So it is going to form four cells. These four cells is called as microspore tetrads. Microspore tetrads from the PMC, and is known as microsporogenesis. So this process is called as microsporogenesis. How many? What is the division? Meiosis. How many meiosis? Only one. Meiosis only one. One meiosis. So meiosis one, meiosis two. This is altogether comprises only one meiosis. Right. So the development of microspore tetrads from the PMC is known as microsporogenesis, right? Late, the microspore tetrads separate and result the pollen grains. The ploidy of the pollen tetrads or the pollen grains is haploid. Because it has undergone reduction division meiosis. Okay. Each microsporangium produces several thousands of microspores or pollen grain. So each microsporangium, sporogenous tissue is like this and is composed of many cells like this. It's a homogeneous tissue, so all cells are identical, right? So now each cell is becoming MMC. So each cell of this is becoming MMC, or you can call it as PMC. Now this is undergoing meiosis and each one is producing 4 pollen grains. So assume there are 40 cells. These 40 cells of this sporogenous tissue is differentiating into MMC or PMC. Then how many pollen grains will be formed? Means 40 into 4. So 160 pollen grains you will get. Right. The ploidy of the is haploid, right? Each microsporangium produces several thousands of microspores or the pollen grains because the homogeneous tissue is composed of several thousands of cells. Now we observe these diagrams. So this is a pollen grain. This is the pollen grain structure. This pollen grain structure, it is this. So the outer layer is called as exine, followed by intine. The nucleus cytoplasm is there. Then it is slowly becoming vacuole. A vacuole is coming out. Vacuole is forming. Then asymmetric spindle is forming. Then it is dividing into two cells: vegetative cell, the generative cell. The vegetative cell is very large. The generative cell simply is floating on this vegetative cell. Right. The generative cell is floating on this vegetative cell. So this diagram you will understand the next page. So pollen grain is a male gametophyte. Okay, each pollen grain is a male gametophyte of the angiosperm. Very, very important statement of the angiosperm, right? It is spherical and measuring the diameter of 25 to 50 micrometers. Pollen grains consist of exine and intine wall layers enclosing a cytoplasm and the haploid nucleus. Haploid, what is haploid? En. Okay, pollen grains are architectured in various patterns and designs. So it is a typical pollen grain. Outer layer is exine. It is very hard, intine, followed by vegetative cell and generative cell. And this is a microspot diagram. And this is scanning electron micrographs of the few pollen grains. So these are obtained from electron microscopes. Diagrams are equally important. Right. Exine is outer spiny and hardy layer. So you could have seen this exine is outer spiny, how spiny it is. Outer spiny and hardy layer exine, right? Composed of a complex substance called as sporopellenin. So this exine is composed of sporopellenin. This sporopellenin is highly resistant to temperature, strong acids, and even strong alkali. At certain regions, the exine is thin and is known as germ pores. So germ pore is not indicated in this diagram. I will show you. So it is like this. Now, so this is exine. Its outer spiny and hard layer, but at this point, the sporopellenin is missing. This hard covering sporopellenin is missing. So this pore is called as germ pore. This pore is called as germ pore. At certain regions, the exine is thin and known as germ pores, where sporopellenin is absent. The hard sporopellenin containing exine helps the pollen grains to preserve well as a fossil. Intine grows out through the germ pore region as pollen tube. So it's an intine layer. So it's a germ pore. Now intine is going to form pollen tube. Intine is going to form pollen tube. Intine grows out through the germ pore region as pollen tube during the germination of pollen grains on the stigma. So after attaching the pollen grain to the stigma, this germ pore leaves a space for intine to come out as pollen tube. Intine is inner thin layer made up of cellulose and pectin. Intine is thin layer, very very thin, but exine is hard layer, right, of cellulose and pectin substance. Cytoplasm of pollen grain is enveloped by plasma membrane and also possesses the haploid nucleus. Certain plant pollen grains are allergens and cause asthma, bronchitis, and other respiratory problems. The carrot grass plant, Parthenium hystrophorus. The carrot grass plant, Parthenium hystrophorus, is causing allergies like dermatitis. Dermis means this is related to skin. The doctor who sees this are called as dermatologist. And respiratory malfunctions in humans. Pollen grains are enriched with nutrients, hence are called as pollen tablets and syrups. The pollen food supplements are found to be effective in increasing the performance of athletes and racehorses. So you could see the diagram of syrups bottle B, and it's a tablet, pollen tablets. Okay. Pollen viability varies from few minutes as 30 minutes in rice. Rice and wheat are collectively called as cereals. They are monocots. From angiosperm. From angiosperm. Right. To several months. Its viability means its ability to fertilize. Ability to fertilize. The the which one? So pollen grain is going to fertilize egg cell. Right. So this viability in several months in case of family Rosaceae, Leguminaceae, Solanaceae. Okay. Pollen grains are stored in liquid nitrogen gas minus 196 degrees Celsius and is known as pollen bank. So it is stored in liquid nitrogen minus 196 degrees Celsius and is known as pollen bank. This is a neat 2018 question. At what temperature the pollen grains are stored in liquid nitrogen minus 196 degrees Celsius? Right. It is known as pollen bank. It is ex-situ conservation strategy. Ex-situ means not in the natural habitat. Right. So male gametes develop within the pollen grains and hence the pollen grains are known as male gametophyte of the angiosperms. Develop within the pollen grains. On maturation of pollen grains, it shows vegetative cell and generative cell. On maturation of pollen grains, it shows vegetative cell and generative cell. The diagram you have seen here. It shows vegetative cell and generative cell. The diagram I want to show to you. Zooming it out. So vegetative cell and generative cell. You could see the generative cell is floating in the vegetative cell. In vegetative cell, the cytoplasm is very abundant. Now you read this. The vegetative cell is large and has abundant stored food, and large irregularly shaped nucleus. Irregularly shaped nucleus. The nucleus in the vegetative cell is irregularly shaped. A generative cell is spindle shaped with a dense cytoplasm. The generative cell is spindle shaped with a dense cytoplasm. Again, you see this. So it is abundant in vegetative cell, abundant cytoplasm. The generative cell is spindle shaped and is having dense cytoplasm. Cytoplasm is having more density. Okay, irregularly shaped nucleus. So genetic cell is spindle shaped with dense cytoplasm. Over 60% of the angiosperms, the pollen grains are shed at this two-celled stage. What is the two cells? Vegetative cell, generative cell. So 60% of the angiosperms are going to release pollen grains in this two-celled stage. Two-celled stage, vegetative cell, generative cell. The generative cell floats in the vegetative cell. I was stressing or emphasizing this point from the beginning. If the generative cell give rise to two male gametes, soon after its maturation, then the pollen grains are shed at three-celled stage. That is, so out of this two cell, vegetative cell, generative cell, this generative cell is going to undergo one mitosis to form two male gametes. Two male gametes. Remember, pollen grain is a spore. Spore means haploid. This is going to form gametophyte. It's going to form gametes, gametophyte, and thus producing gametes, and thus producing gametophyte, right? Two gametes. This generative cell is going into form mitosis and forming two male gametes, right? But 60% of the angiosperm is going to shed this pollen grain at two-celled stage. But they will form this three-celled stage when the pollen grain, upon reaching the stigma, the intine gives out the pollen tube through the germ pore. So after attaching with this, the pollen grain is having two cell, right? Vegetative, generative. This generative cell is going to form two male gametes. Two male gametes. These male gametes is going to be released in this tube. 60% of the angiosperm is doing like this. 40% is doing at this stage itself, anther filament stage itself, they're going to release in this form. They're going to dehyse the pollen grains in this form in 40%, whereas in 60% it is released in this VG form, and after attaching with the stigma, that is after pollination, it is dividing generative cell is dividing into two male gametes. The, the The formation of male gametes from the generative nucleus is known as male gametogenesis. From the generative nucleus, very very important, not vegetative. Mature anther has two chambers due to disorganization of the wall layers between the two microsporangia of each anther lobe. So mature anther has only two chambers due to the disorganization of the wall layers between the two microsporangia of each anther lobe. When the pollen matures, the anther breaks of the pollen grains are liberated through the stomium of the anther wall. The pollen grains formed inside the anther from microsporangia is going to be released. That is, I will show the figure. Yeah. Now this is called as stomium. Here also this is called as stomium. The point where the pollen grains are released are called as stomium. Pollen grains are liberated in the form of yellow dust for pollination. Yellow dust. In case of gymnosperm, pinus will produce sulfur shower. Sulfur shower. This question is asked from plant kingdom lesson, right? Just recalling a recap. So that's all about the male reproductive structure of flower. Now tell me, to, for the formation of pollen grain or the formation of male gametogenesis, how many meiosis, how many mitosis? One meiosis. One meiosis and two mitosis. One meiosis and two mitosis is required. Now the question I will ask you, for 40 pollen grains, how many meiosis should have occurred or should have happened? For 40 pollen grains production, how many meiosis should have happened? What should you do? So 40 pollen grains are produced. You know that through one meiosis you are going to get 4 pollen grains. One meiosis is going to produce 4 pollen grains. Then 10 meiosis is going to form 40 pollen grains. So 10. Actually what you are doing, Number of pollen grains by 4. This is the formula you have to use. Number of pollen grains given by 4. In case of female, use the n itself. That will be done in the next session. Till then, stay tuned. Like, subscribe, share. Thank you.